law of dominance. The word itself says it is about dominance, meaning one character of a plant is dominating the other character. These characters that are present in the parent, they segregate from each other such that a gamete receives only one of the two factors. The color gets separately assorted in the progeny and the shape of the seed separately gets assorted in the progeny. So that is what law of independent assortment states. Hello everyone, a warm welcome to this session of concept class for class 12 biology. I am Dr. Divya, biology faculty. So in this session of chapter 4, that is principles of inheritance and variations, we will learn about Mendel's law. So in the previous session, we had learned about Mendel, that is how he chose his plant, with what characters did he choose his plant, and also we got to know about the monohybrid cross, which spoke about the law of dominance and also the segregation and all that. So in this session, we'll concentrate on all his laws and also we'll talk about the dihybrid cross. We'll concentrate on all the laws, that is the statements that are there under this particular laws, and also we'll learn about the law of independent assortment, wherein taking dihybrid cross of the pea plant as an example. So talking about Mendel's principles or Mendel's laws of inheritance. So his first law, which is called as law of dominance. The word itself says it is about dominance, meaning one character of a plant is dominating the other character, or one factor, according to Mendel, who he called those characters as factors. One factor is dominating another factor, or one gene is dominating the recessive gene. So here characters are controlled by discrete units called as factors. I had told you about this in the previous session itself, right? During those times, that is when Mendel first started his experiments on the pea plant, he didn't call those as genes. Instead of calling them as genes, he gave the name as factors. Later on, after the inventory of the microscope and all that, they got to know about the chromosomes, the genes and all that. But until then, it was called as the factors. There are something in the plant which are called as factors that control all the characters of the plants, is what he means. And these factors, they usually occur in pairs. They are always present in pairs. Say, for example, we'll consider the characters which are controlled by the characters that I'm talking about now. Is we'll take the example of the tall plant and the short plant, right? So tall plant should be expressed by some factors. So we'll take for tall plant, it is T, and for short plant, it is represented by small t. Now we can't take this and do the calculations. It should always be represented in pairs. So we have to write it as TT, small t if it is homozygous. If it is heterozygous, it will be a capital T and a small t. So it is like this, we need to express them, because factors, according to Mendel, always they occur in pairs. So that is why. And in a dissimilar pair of factors, one member will be dominant and one will be the recessive. So this will be the dominant one, this will be the recessive one, and always it is the dominant gene that gets expressed. It is according to Mendel's law of dominance. So always the dominant gene, say for example, when we make a cross between the tall plant and the short plant, we'll always get the tall plant, because the genes responsible or the factors responsible for the tallness of the plant is always one hand upper, that is, it is dominant. So that is what this law means, that is law of dominance. So next we'll move to second law, which is law of segregation. Now the name says segregation. What do you mean by segregate? It means to separate. So here, during gamete formation, the factors. Now what are the factors? They are nothing but alleles. So what are alleles? Alleles are nothing but these are alleles. So all these are alleles which are present in a chromosome. So they are nothing but alleles. So genes. So on the DNA, we find genes which are always in triplet, like ATG, then ACG, like this. They are all in triplets. Now this is one gene, this is one more gene. Now among these genes, one which is called as the factor or the allele, that is responsible for the character of that particular individual. Say for example, one allele will be responsible for the eye color of the individual, one allele will be responsible for the hair color, or in a plant the allele will be responsible for the flower color, the allele will be responsible for the tallness of the plant, for the plant to be dwarf, all that are nothing but called as alleles. So during gamete formation, the alleles of a character pair present in the parents, they tend to segregate. Say for example, in this there is one allele, that is T. So this is present in the male parent. Say for example, this is present in the female parent. They have TT. Now when we cross, what will happen? We'll get T, then TT. So like that we'll get the characters. So all these characters, they tend to segregate. So we saw that, right? While doing monohybrid cross, we got to know that. So you have a look at that particular session, that is session 1. So you'll understand it better. So whatever characters are there in the parent, they get segregated and they get expressed individually in the progeny is what this particular law means. And these characters that are present in the parent, they segregate from each other such that a gamete receives only one of the two factors. Say for example, the progeny, it can't receive all the factors, right? Either it can receive a factor which is responsible for the tallness of the child. So whichever is dominant, that factor it will receive. Homozygous parents produces similar gametes. That is true, because homozygous parents produce a similar gamete means, say for example, this is a homozygous crossing between a heterozygous. Now they'll have the similar gamete, right? So they'll have T and they will have T here. So homozygous parents will have similar gametes. Heterozygous parents produces two type of gamete. That is right? Heterozygous means TT. It will produce which two type of gamete? One of the gamete will be T, other gamete will be small t. So it will produce two types of gametes, each having one allele with equal proportions. This is also important. So in heterozygous, but which will get expressed, it is the tallness is the one that gets expressed according to Mendel. Later on, all this was proved wrong because intermediate characters and all that will come. We'll study about that in the later classes, while that is in the next session, we'll get to know about it in codominance and incomplete dominance. So this is about law of segregation. That is, whatever characters are there in the parent, they segregate and they get expressed separately. Next is talking about the third law. So those first two laws which I told you now, we can take the example of monohybrid cross or inheritance of one gene, wherein in that particular cross, we also saw the law of dominance, wherein the tall plant was dominating the short plant and the tallness factor was getting expressed in the progeny or in the F2 generation. And in also we learned law of segregation also. For that also the same example holds good, because the characters tends to get segregated, where in the F2 generation we will get tall plant, we will get short plant, we will get homozygous tall plants, we will get heterozygous tall plants, we will get homozygous tall plants. So segregation of the characters will be found there. So for this law, that is Mendel's third law, which is called law of independent assortment. So here also, when we take a dihybrid cross, we have different characters there. Say for example, we have the color of the pod, the shape of the pod. Now, we are crossing between two pea plants, wherein we have considered two characters. One is color of the pod, and in both of the pea plant, we have considered two characters, color of the pod and the shape of the pod. Now, when the progeny is formed, both of these characters also will go into the progeny. There also we need to consider the color of the pod as well as the shape of the pod and all that. So therefore, the characters tend to get independently assorted. So assortion means we are separating. Say for example, we have marbles of different color, right? Now you assort them. It's called assorting. Segregation means you're just separating them. That's it. Assorting means like you have a certain idea in your mind, where in yellow color and all you have to assort and put it in one place. Blue color you have to assort and put it in one place. Assorting means distribution based on the character. Okay. So that is assortment. So here, when more than one pair of characters is involved in a cross, say for example, I gave you the example of the color of the pod and the shape of the pod. Two characters we are considering here, right? That is it. So when more than one pair of characters is involved in a cross, the factors will pair independently, segregate from the other pair of characters. Now, this you will understand when I explain about this. That is inheritance of two genes in dihybrid cross. So this is one of the best examples to explain Mendel's third law. Which is that third law? That is the law of independent assortment. How the characters get assorted and how they express themselves in the progeny is what we are studying here. So a cross between two parents differing in two pairs of contrasting characters we have to choose here for dihybrid. Now the name itself says it is called dihybrid. Here two characters are chosen. Okay. In monohybrid, only one character was chosen. What was that character? It was the height of the plant, wherein we took tall and short, right? Here two characters are being chosen. So what are those characters we will look into? So Mendel crossed pea plant with the shape of the seed was taken into consideration, shape of the seed, and also how the seed looked. That also was taken into consideration. So what was considered here? Shape was considered, color was considered. In shape, what did he choose? He chose round and wrinkle. In color, what did he choose? He chose yellow and green. Now, when crossing over takes place, when we do crossing over, no, don't think one seed will be round and it will be wrinkled. No, characters, it will get assorted separately. That is, either it will have round yellow seeds, wrinkle yellow seeds, round green seed, or wrinkle green seed. So that is called as assortment. Now you understood, no? So it will get assorted. So next, talking about inheritance of two genes, that is dihybrid cross. So in dihybrid cross, the name says di, meaning two characters are taken into consideration. So what are the two characters that are taken into consideration? One is the shape, right? Shape of the seed. The other one is color of the seed. Now, shape of the seed, what all they took? Two shapes that were considered. One is round. So, round was represented by capital R. One more shape they considered was wrinkle. Wrinkle was represented by small r. Then, color that they considered was yellow. Yellow is represented by capital Y. Next color is green. Green is small y. Now here, which is the dominant one? Round and yellow are dominant. Recessive is wrinkle and green is recessive. Now we know so much. Okay. So now we are assorting the characters. So when we assort the characters, no, we'll not have a plant which is having round wrinkle seed or yellow green seed. No. The color gets separately assorted in the progeny and the shape of the seed separately gets assorted in the progeny. So that is what law of independent assortment states, because each character independently they get assorted into the progeny or to the F2 generation. So here we'll consider this. So what we'll do here is we'll take the male plant and we'll cross it with the female plant. So now these are the parents, right? Parents, what are we taking now? We are taking round yellow, crossing it with what? We are crossing it with wrinkle green, crossing it with wrinkle green. So round yellow, it is represented as R, Y. Wrinkle green is represented as small r, small y. Now, what will be the gametes here? So, we have got in the F1 generation, what did we get? We got R, R, Y, Y. So, what did we get here? We got a plant that is producing round yellow seeds. Now, we'll go in for selfing. So, here also, which are the parents that we have chosen now? These will be the parents. The F1 generation will be the parents. So, it is R, R, Y, Y into R, R, Y, Y. So, these were the gametes for this. Now, for this, what are the gametes we will write? So, for this, it will be capital R and Y will be one gamete. Capital R and small y will be one gamete. Small r and capital Y will be one gamete. Small r and small y will be the other gamete. Now, we have to put the Punnett square for this. So, that is what I have put here. Now, wherein male is capital R, capital Y, female is also capital R, capital Y, because the characters are getting assorted. What do we get after putting all of this? We will get here round yellow. Again here also round yellow. Here again it will be round because according to first law of Mendel, the dominant character will get expressed. So therefore here round, yellow. Here also again round. This is yellow. Now here again it is round, yellow. Then here again it is round, but it is small y. So small y means what? Small y is for green, right? So it is green. If it was capital Y, small y, it would have been yellow only because again their dominant character will get expressed. Say for example here, it is round, yellow. Now here it is round, green. Here again round, yellow. Here again round, yellow. Now here it is wrinkle, yellow. Here again wrinkle, yellow. Again here round, yellow. Here round, green. Wrinkle, yellow. Here it is wrinkle, green. Now after this we need to calculate the phenotypic and the genotypic ratio. So talking about phenotypic ratio, what all phenotypic means, as you see, we'll get to know which plant is having round yellow seeds, which is having round green seeds, or which is having wrinkle yellow seeds, wrinkle green seeds. What it will be here? We have to consider round yellow. We have to consider round green, then wrinkle yellow, then wrinkle green. So much we have to take into consideration. So let's start counting. So 1, then 2. Which one? For round yellow. Okay, I've taken purple. So 1, 2, 3, 4, here 5, then we have 6, then in the 7, round yellow again, 8, one more round yellow, 9. So what is it? 9 is there. Next, round green. So how many round green shall we count? So we have 1 here, then 2, then round green, 3. How many? 3. Sky blue for wrinkle yellow. So wrinkle yellow will just count here, 1, then 2 and 3. So it is 3. Now wrinkle green. So wrinkle green, it's black. So how many? Only 1. So it is 1. So the ratio is 9:3:3:1. So this is phenotypic ratio. Counting genotypic, it's quite laborious because too much we have to look into carefully. We have to look into homozygous and heterozygous and count, wherein how many RRYY is there. Okay, so I'll write it as how many RRYY is there we need to count. And we need to count how many RRYY is there and how many RRYY is there, and again capital R, small r, capital Y, small y. So many I've already written here. How many RRYY is there? How many RR capital Y small y? How many RRYY? Here all the homozygous, heterozygous separately we need to take into consideration. So the ratio for this will be, for genotypic ratio it will be 1:2, 2:2. This if you don't write in the exam genotypic ratio, it's also fine, but you need to remember it. 1:2:2:4:2 1:2. So this is the phenotypic ratio. So, what all did we take? RR, YY, then RR, YY, then RR, YY. So, what all did we take? RR, YY, then 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 RR, capital Y, capital Y. So like that we need to take and we need to count this. So this is about the dihybrid cross. So this is one of the best examples for law of independent assortment. The other two, that is monohybrid cross, is one of the best examples to take for law of segregation and law of dominance. Here also we can see law of dominance. For law of dominance and law of independent assortment, we can see here as well. So this was about the session. In the next session, we'll talk about non-Mendelian inheritance. So apart from Mendel also, there are some genetic experiments which were conducted and they were called as non-Mendelian inheritance. So we'll see which proved that Mendel's concept is not completely right. So we'll talk about incomplete dominance and we'll talk about co-dominance. So we'll talk about these two in the next coming session, taking examples and learning about this. So see you in the next session. Thank you.